Buenas tardes. Vamos a comenzar con esta parte, con esta conferencia Ruben Budelli. Mi nombre es Ana Silva y soy encargada del Laboratorio de Neurosciences de Facultad de Ciencias, y desde allí hemos tenido esta iniciativa de presentar la conferencia anual Ruben Budelli, que esta es su segunda edición. Una conferencia que honra a quien fue el primer director del Laboratorio de Neurosciences de nuestra facultad, y que es una actividad destacada en las jornadas de la Sociedad de Neurociencias, también auspiciada por la Federación de Sociedades Latinoamericanas, FALAN. Esta va a ser como ustedes verán, una conferencia híbrida. O sea que tenemos al mismo tiempo la audiencia presencial, nuestra conferencista que está hablando desde la Universidad de Colorado en Estados Unidos, y también es seguida por una audiencia virtual a través de los canales de YouTube y el Zoom que generamos a través de FALAN para este evento. Quiero entonces agradecer especialmente la presencia de nuestra decana interina de Facultad de Ciencias, la profesora Ana de Nicola, que se dispuso a dar unas palabras para dar comienzo a esta conferencia.
Bueno, buenas tardes a todos. Para mí es un placer estar acá en la inauguración de la conferencia Profesor Ruben Budelli en su segunda edición. Y bueno, y en nombre de la decana Mónica Marín y de toda la Facultad de Ciencias, queríamos decirles que celebramos esta iniciativa de la Sociedad de Neurociencias del Uruguay de homenajear al profesor Ruben Budelli con una conferencia que lleva su nombre. El profesor Ruben Budelli fue un destacado docente de nuestra Facultad de Ciencias, con un fuerte compromiso por la institución y un desarrollo y contribución al desarrollo de la ciencia del Uruguay, con un fuerte foco en la interdisciplinariedad. Entonces, sin lugar a dudas, esta manera hermosa es una manera hermosa de homenajearlo, de difundir su pensamiento y llevar el legado de Ruben Budelli a mucha más gente. Así que muchas gracias, los felicito.
A continuación le doy la palabra a Daniel Herrera. Daniel Herrera es asistente del Laboratorio de Neurociencias, actualmente haciendo su postdoc en UPenn, en Filadelfia, Estados Unidos, y es con quien hemos coordinado esta iniciativa. Él les va a presentar lo que significa la conferencia.
Gracias, Ana. Bueno, gracias a todos por acompañarnos en la segunda conferencia anual Ruben Budelli, y gracias a la SNU también por darnos este espacio tan grandioso. Bueno, estamos nuevamente celebrando la memoria de Ruben, y ya se dijo bastante de la importancia que tiene Ruben para la neurociencia en Uruguay. Para los más jóvenes, Ruben probablemente haya sido tutor de sus tutores o haya estado en sus tribunales de tesis. Pues unas breves palabras sobre Ruben. Yo creo que es justo decir que si le hubieran dicho a Ruben de hacer una conferencia con su nombre, hubiera estado muy opuesto a la idea, ¿no? Por una cualidad suya que puede ser de humildad o también como una aversión a la solemnidad que tenía. Pero para homenajearlo también a Ruben le encantaba discutir, como saben los que lo conocieron. Así que, aunque no le hubiera gustado la idea, me parece que llevarle la contra también es una buena manera de homenajearlo. Creo que Ruben hubiera disfrutado mucho la oportunidad de una jugosa tertulia en torno a la idea de nombrar a la conferencia Ruben Budelli. Fui de las últimas generaciones en conocer a Ruben, y una cualidad suya que recuerdo con aprecio es que empezando mi doctorado, mi maestría, Ruben me hablaba de igual a igual, aunque estuviéramos en las antípodas de la carrera científica. Y eso me hacía sentir parte de una larga tradición científica en nuestro país. Entonces, ya que hay una audiencia tan grande y multigeneracional, destaco esa cualidad integradora que tenía Ruben, y también darle la bienvenida a las nuevas generaciones y bueno, aprovechar esa memoria para instar a los más jóvenes a que se animen a hacer preguntas y sacarle charla a los más viejos a lo largo de la conferencia y que disfruten esta excelente charla que tenemos por delante. Así que ahí la hago corta y ya le paso la posta a Ana.
Bueno, y ahora voy a introducir a quien será nuestra conferencista. Y para eso I will switch to English, okay? So, I am really honored to introduce Professor Tracy Bale as speaker of this second edition of the conference Ruben Budelli. Tracy Bale is the Anschutz Foundation Endowed Chair in Women's Integrated Mental and Physical Health Research and the Luttman Center and Director for Intergenerational Stress and Health and for Sex Differences Research in the Department of Psychiatry in the School of Medicine of the University of Colorado. She completed her PhD at the University of Washington in the Department of Pharmacology and her postdoctoral work at the Salk Institute with Dr. Willie Bale. Dr. Bale was professor of neuroscience at the University of Pennsylvania for 15 years prior to her move to the University of Maryland in Baltimore, where she was recruited to develop the Center of Epigenetics Research in Child Health and Brain Development in 2017. Her research focuses on understanding the role of stress dysregulation in neurodevelopmental and neuropsychiatric diseases and the sex differences that underlie disease vulnerability in humans using the mouse as a preclinical model. Her work has been recognized all over the world, and she has recently become a member of the National Academy of Medicine in the USA. But Tracy is way more than her science. She has been the president of IBRO since 2020, and as the first female being IBRO president, she has revolutionized IBRO towards transparency, inclusion, and diversity. This is a very important thing because in this audience today, we have most of the regional committee of IBRO members and also all the neuroscience community that has a lot to do with this IBRO commitment towards the progress of neuroscience in Latin America. It is also very important to homage the legacy of Ruben Budelli by this big person as Tracy is, because he himself pioneered the search and establishment of regional and international collaborations in Latin America and in the world. So thank you so much, Tracy, for accepting this invitation, and the floor is yours.
Daniel, can you hear me? Yes, okay, great, thank you. So first I want to start off by thanking Anna and Daniel and for the invitation from the Society to participate. It's a true honor, and okay, I can see some people in the audience, that's fabulous, thank you. And it's a real privilege to be giving this very special lecture and to really, I think, connect globally, which we all, I think, have a great desire to do, to bring the world closer, to elevate neuroscience, both access to resources, advocating for trainees especially. I hope we have a great number of trainees in the audience. I'm squinting to see. And I also want to give a big shout out both to our IBRO regional committee, Latin American regional committee members. Maybe they can show their hands. I can't see them. Jose, are you there? Sorry, yeah? Yeah. Okay. And I also would very much like to give a huge applause to Falan. Also, it's been a great privilege to, over the years, have the opportunities to work with. I'm trying to see if I can see Carolina, who's all there. I wish I was there because I have to tell you, here in the great state of Colorado, it is a snowstorm, and we already have 2.5 feet of snow on the ground, and more is coming down. So I do wish that I was there with you in person to share the great joys of neuroscience. I hope that I can speak to the breadth of neuroscience as I give this lecture, and I also want to make sure that I'm paying a great respect to Ruben, as I know you heard earlier in the introduction, to his contributions to global neuroscience and advocating for trainees' inclusion and diversity. When I was doing a search for some images to pull up, my favorite is really this one on the bottom here, that somebody did this incredible sketch, and I couldn't find the authorship. So if you're in the audience, congratulations to you. And I think paying respect to individuals who have really dedicated themselves in the great world of neuroscience to ensuring that trainees get the support and access to the resources, and that we really speak to how powerful inclusivity globally is for all of our neuroscience. And I know that Daniel is doing a postdoc at Penn, which is an exciting place to be. I really enjoyed my 15 years there. And I hope all of you have opportunities to access neuroscience, whether it's through mentorship or trainees have access through IBRO programs to travel. I do think that the more we interact, the better all of our neuroscience and research becomes. And so I'm happy to answer any questions at the end that anybody has. I know we have, like I said, our regional committee members there in the audience that hopefully can answer questions as well about neuroscience. So speaking to my research, I think you heard in the introduction that I am a neuroscientist, but my background is in pharmacology as well. But I am really interested in brain development across psychiatric. I'm interested in the brain and how its development can be altered by the environment. So that could be the preconception environment, that could be pregnancy itself during early brain development, that could be the postpartum adolescence. Your brain continues to be affected, of course, by the environment that you live in. But understanding when and how those effects, I specifically study stress, or in humans, we study adversity or trauma. I do both preclinical and clinical research, and we're very interested in trying to help better understand neuropsychiatric disease. So stress is one of the most common pervasive effects that is a risk factor for presentation of all neuropsychiatric disorders. We all experience stress. I will say, as an American, this last few months have been incredibly stressful in the US, and the election results are even more stressful, but we can come back to that. The stress that we're interested in is not the acute stressors, a final exam and a course, submitting a grant, those kinds of stressors, but rather the kind that is pervasive and compounding and is important across the life course. Here in the US, and I know you as well, have experienced the political divisiveness that is a daily compounding stressor. Many of you, probably especially the younger members and trainees in the room, I think are very stressed by keeping up on social media. There are, of course, very positive aspects to social media that connect us all, and we can use as markers of advocacy for ensuring that people around the world know about the incredible neuroscience that's going on in Latin America, or that's going on in Africa, or that's going on in Asia. Social media is how we connect. However, it can also be a source of great stress, trying to keep up, feeling like everybody is doing so well and perhaps feeling like you're not. And I think that can be a source for, especially those individuals who grew up with social media. I think we're seeing a lot of effects of that. Global and climate change huge source of stress for many individuals around the world, facing everything from fires and floods and tornadoes and hurricanes, et cetera, droughts. This is another source of incredible stress that we live with. The pandemic, we all know, was an incredible lengthy stressor of unpredictability, not knowing what was going to happen, how it was going to happen, how we were all going to survive, and the outcomes of that. I think I want to take a second just to give a reminder to everybody who is, senior faculty in the room, to remember as our more junior faculty come up for reviews, tenure processes, promotions, that the pandemic was such a difficult time for starting labs, hiring people, getting equipment, getting resources, that I don't want us to forget as those individuals go through the promotion process that was such a stressful time. And then we have the US election, which I will say personally continues to be an enormous stressor that is not going to go away. We hope in 4 years it might go away, but I'm doubting it. I think we remain to see what this effect has on the world, what effect it has on research. People in the US are already panicking as to what this presidency and this administration may do to science and medicine, what it may do to NIH, which we know funds all of the research within the US. There's a lot to unpack. Anyway, I just think it's important as a consideration that I want to highlight that it is a concern. I think it's being discussed at many different levels. And so, anyway, I'm going to move on from this because it is a very stressful point, and certainly within the US, but I think around the world. These are statistics following the pandemic that I'd like to discuss because I think it speaks to the fact that stressors, especially those in chronic in nature, can be very long lasting. These are statistics of American adults presenting with anxiety, depression, or anxiety and depression. And those graphs there, you can see going through December of 2021. I'll show you in a minute some more recent data. I like this data because what it shows you is the high percentage of adults, so over the age of 18, presenting with neuropsychiatric disorders compared to where we were before the pandemic. And that's the dashed line going across there. So somewhere between 8 and 10%. We're now well over 30%. And I will tell you, on the campus where I am, the professional medical campus, so it's all students who are of, I would say, somewhere between an average of 24 to 27 years of age, that if you look at the statistics, 67% of our students, adults, are currently taking a neuropsychotropic medication for anxiety and depression. Okay, I'm just going to start talking again unless someone tells me not to. So this is the most recent data that's done a thorough assessment within the US following the pandemic. And I just wanted to highlight across adults. They've also broken this down by age. And you can see again, as I mentioned, that young adults here broken down into 18 to 24 years of age, 50% are currently presenting symptoms of anxiety or depression. And so the effects of the pandemic, because of its chronic nature, for individuals who may have been at risk for a neuropsychiatric disease, we're definitely seeing a much, much higher and lasting effect of the pandemic. I don't think that's terribly surprising, and it's probably globally being seen with our current mental health crisis. I also want to add in there a shout out to thinking about vulnerability to mental health disorders, consideration of the effects, the chronicity of effects of discrimination, racism, poverty, and violence, as defined here by the US Surgeon General, that these stressors on these diverse ethnic racial groups are also pervasive and can also be a huge risk factor. And sadly, also for groups that usually have the least access to resources for treatment. Daniel also wanted me to talk a little bit about the work that we do outside of the lab. So I just have a couple slides here to give a shout out and a reminder that it is so critical that we as scientists engage with our communities. It is, I think, because, and I'm going to speak largely about what has happened in the US, but I think it's also global. that when we, as the most educated and trained scientists and physicians in our institutions doing our work, but not engaged in our communities, what has happened is that we've created a bit of an isolation. And in the US, I think as around the world, this has resulted in a really negative perception within communities about being elitist. That's a huge effect here in the US. And when people don't trust or don't understand what is happening, they tend to doubt it. And this has led in the US to an enormous distrust of science and medicine, that it's no longer just given as fact. Conspiracy theories abound. People are less able to critically think about what is being said. And quite honestly, has led to all of the election of the Trump administration, as well as the taking away of women's reproductive rights largely across the US. And I think we have to, as scientists, really take some responsibility and make more of an effort to communicate in a non-judgmental way with our communities so that they understand the processes. How are vaccines made? How are they tested? How can we be more engaged in our communities to win back that trust of process? One of the ways that we do this, my lab does a lot of work in the community across different areas. So we do work with communities, starting with children. And I think this is one of the easiest ways to begin to formulate that relationship that builds trust. When you start with children in schools and programs, we run a camp that talks about the brain and awareness of mental health, et cetera, with children, second, third, and fourth graders in the summer. We do a lot of fun activities, hands-on science, which every child loves. But we also do a lot of creative ways of teaching. We do it through the science, we do it through art. And it's through those processes that the kids become very excited about the brain. And of course, when they go home, then they talk to their families, and then their families engage in the communities. And so it's a really, I think, impactful way to engage in our communities. We have a lot of family members, a lot of parents that come into the camp and ask lots of questions and are very excited about the opportunities. Oftentimes that leads to other relationships where those family members may then ask additional questions. Could you come talk to this group? Could you come and answer questions for another group? When I was in Baltimore, it led to participation of the mayor's office, which then led to interactions of city council, which are the policy makers for the community in the city. And that led to establishing some really great, significant relationships, building an understanding about the brain, building an understanding about the effects of trauma, racism and discrimination effects on the brain. So this is just my way of saying anything that you can do, be it big or small, is impactful and important. And you don't have to have something professionally organized. I always just say, just start with something. My experience is that if you just start with something small, it very often leads to other relationships in the community that are both exciting. I think as a scientist, we always have these I think strong drives as scientists to help educate and communicate and listen in our communities, and I think it's always a win-win. So that's my suggestion for everybody, that maybe as a New Year's resolution, we think about that. So in thinking about brain vulnerability, I often like to go back to the basic concept that I'm sure is familiar to you all, which is the G by E by D. And that is, the G is the genes that we all inherit from our parents. Can't do much about the DNA that we inherit. And we all know that within families, there are risk. And those risk and resilient factors could put us at risk for obesity, diabetes, hypertension, metabolic disorders. They could put us at risk for mental health disorders. They could put us at risk for cancer. Understanding the genes through a lot of effort in GWAS studies is informative. It helps us understand the genes that are involved. Sometimes it points in directions of drug development or interventions. We lack a lot of this understanding in neuroscience and psychiatry. We're just really beginning to tap into some of this risk and resilience. The E is the environment, and we all appreciate and understand that the influences of the environment, whether it's preconception alterations that mom and dad bring together from across their lifespan that can influence signals at conception. It can be influences of mom's pregnancy, metabolic stress, trauma, etc. And they can be influences across the rest of the lifespan that continue to determine our risk and our resilience. The D is developmental windows. And those are when those environmental, both positive and negative, can occur across the lifespan. So that can, as I've highlighted, be preconception, that can be during pregnancy, adolescence, young adulthood, perimenopause, etc. That those are what we call developmental windows. At the bottom is SABV, and that is sex as a biological variable. And sex as a biological variable is significant, and it intersects at all three of these levels. So the genes on the X and Y chromosome can be very impactful for rates of development, transcriptomic regulation. There's lots of really interesting differences between XX and XY, and a lot of genes, for instance, on the X chromosome that may escape X inactivation, being very protective, for example, in utero for females as they develop. SABV determines a lot about how we perceive our environment, how society may determine gender interactions with our environment. And of course, sex as a biological variable is very important for developmental timing and experiences, especially as we think about pregnancy, postpartum, and the adolescent and pubertal windows. So all of those factors are things that we consider in my lab as we think about how the brain and changes in development. But one of the things we lack profoundly in psychiatry, unlike other diseases. So for instance, cancer, we have lots of biomarkers for cancer. So if you're experiencing symptoms and you go into the physician, there's lots of ways if they suspect cancer that they could test by looking at blood markers or imaging. The same is true for metabolic disorders. If you are pre-diabetic, for example, they could look at your A1C levels. Unfortunately, for psychiatry, for understanding mental health, we don't have really any solid biomarkers in the way that we can really identify biologically the disease or the disorder that someone is at risk of or presenting, or avenues to tell us to determine which sorts of medications would be most effective. One of the ways in my lab that in the last 15 years we've begun really thinking about biomarkers is thinking about the organism as a whole. So this means not just thinking about the brain in isolation, but rather thinking about how health may be affecting mental health as a process. And one of the key markers that we've been looking at in the last 15 years are called extracellular vesicles. Extracellular vesicles are nanoparticles, so they're teeny, teeny, tiny particles secreted from all cells in the body, including the brain, and they act on all cells in the body, including the brain, in all mammals. There are different types of extracellular vesicles, which I don't really have time to get into today, but the cancer field has really done the most work in the last 2 decades, assessing the value of these active, very dynamic particles and how they may tell us a lot about the health of an individual and the changes in health conditions that are important for understanding the brain. So these nanoparticles are rapidly secreted, and there's very specific cellular signals. So if you think about the endocrine system and how different tissues may release hormones or factors that act locally or at distant sites, nanoparticles are very much like that, except with much greater specificity. And so the lipid membrane of these nanoparticles contains a ton of very exciting and engaging proteins that we can learn a lot from. Inside the nanoparticle, are additional proteins, parts of mitochondria, and a lot of small non-coding RNA. And it is the process that secretion of nanoparticles or extracellular vesicles, I'll refer to them as EVs, that they travel in very high concentration in circulation. And the time in the lifespan at which they are the highest in concentration, which tells us a lot about their function, is actually pregnancy. And that is because, in part, the placenta is producing massive amounts of EVs that travel in circulation. Their main job is to try to facilitate and maintain homeostasis. And so they are responding to changes in the environment and reacting to them and signaling to different tissues to try to maintain homeostasis. They have both local and systemic functions. They are the big part of their function is cell communication. So they travel locally and at distant sites between tissues. And as I said, they are very dynamic. And we think in part that the different types, so there are larger types of extracellular vesicles called microvesicles, and those are secreted and blebbed from a cell membrane. So when you detect them in circulation and detect differences in proteins, you can isolate which types of tissues might be more or less engaged in a dynamic process. And then there are small, teeny tiny EVs, and those are called exosomes. And those are really the highest concentration traveling in circulation. And those are sort of like a big cargo ship that they are moving ahead, trying to maintain homeostasis, very difficult to turn, so they're not as dynamic, for instance, as microvesicles. So by measuring systemically changes in these EVs, the populations, and their cargo, we can learn a lot about the experiences of an individual across the lifespan by changes in how they dynamically respond to their environment. There's many different ways to look at differences and measure them once you isolate them from circulation in humans or mice or cell culture. I'll show you some studies on all of those. On the left, you can see we look at the average vesicle size. In the center, there is a piece of equipment called a particle matrix zeta view. It's a nanoparticle tracking system. So when we isolate our vesicles, we can put them into this system and it will tell us, it will give us readout of the average size. It'll give us a spectrogram of the size distribution of the EVs. On the right, I'm showing you can also look at the zeta potential, which is the average size, the average charge change. And that, as you can see, is a reflection of change in both size of a particle as well as the protein composition. It has a lot to do with the slipping plane of how particles interact. This machine uses Brownian motion, so it can detect differences in all of these outcomes, including also concentration, which I'll come back to. So these are important because they can tell us very quickly if something is different in an individual. So if we have, for instance, an individual who has had a lifetime of adversity and trauma, and so they are unable to maintain homeostasis, and we can expose them in the lab to an acute stress experience, taking a blood sample before and after the stress, we can look at these changes and make an assessment of whether that individual's cells, and again, we can look at specific tissues as well, and it can tell us information about things like the endocytic rate or the uptake of vesicles, the type of vesicles, and the stability of those vesicles in circulation, all of which is very important for maintenance of overall health. So as I said, we're interested in how stress, trauma, adversity across the lifespan can affect not only the brain's development, but its ability to maintain health. I'm going to tell you two quick stories here. I'm going to try and be fast about this since we're running short on time. Just how we utilize these biomarkers to understand individual health. This study I'm going to first tell you about is a collaboration with Tanya Jovanovic, who is now at Wayne State, Detroit, which is in Michigan. She was previously at Emory, which is in Atlanta, which was called the Grady Trauma Project. So I'll come back to that. So these are a large pool of adult women in their late 30s and early 40s. who had come into the emergency room of an inner city hospital and were recruited over time into what's called the Grady Trauma Study. They capture information about life experiences and when those experiences happened, and they take biological samples and bank them. In our collaboration, we asked a very specific question related to timing specific trauma. So this was related to interpersonal violence or sexual trauma, when that trauma had happened. So we had 4 groups. If you look over here on the trauma questionnaire, you can see that either the sexual trauma happened prior to adolescence, in adolescence, as an adult, or there was no sexual trauma at all. So there's 4 groups of women. There's about an N of 25 in each of the groups. And you can see that as we divided these into timing specific, where that overall these women they're all Black or African American, and they were all overall high levels of trauma in their environment. So we've controlled for that type of trauma. Now we're specifically asking about a very specific type of trauma, interpersonal violence. I'm not going to go through all of the data because it's all published. You can see here in Biological Psychiatry. But what I wanted to highlight is the utility of asking about this specific biomarker. So quickly on the left, you can see that when we assessed these women, first breaking it into no sexual trauma versus sexual trauma on the white and blue bars there, and looking at changes in metabolic outcomes, as many other groups have seen, that women who've experienced interpersonal violence, regardless of when they experienced it, will show higher rates of aging and higher incidence of entering into obesity, diabetes, and hypertension. This is not a surprise. What is interesting is if you break apart the sexual trauma groups into when that happened, you can see that largely this is being driven by experiencing that interpersonal violence prior to adolescence, so childhood trauma. And if you look at the hypertension down below in D, you can see, which also has been shown by many other groups around the world when understanding the effects of trauma accumulation, that these women seem to be aging faster and entering earlier into the hypertension stage 2, or progressing basically faster. And then next would be adolescence, and then next adult. So the earlier and longer you've been experiencing trauma, the faster you age. But what's interesting, when Tanya looked at the outcome for doing fear conditioning and fear potentiated startle in these individuals, I'm just showing you a little bit of her data here on the right. You can see, again, we see the difference in the women experiencing sexual trauma. But if you divide it again by age, this is largely being driven by the exposure during adolescence, so a different time period. So on the left, metabolic outcomes driven by largely childhood trauma, on the right being driven by adolescent trauma. That's not surprising, but what is surprising, when we isolated these extracellular vesicles from these women and blindly isolated the proteins from their EVs, did a mass spectrometry proteomics analysis, as you can see in the middle here, if we first compare all detected proteins in circulation from EVs, you can see some pretty big differences between no sexual trauma and sexual trauma, with red being highly expressed and blue being highly expressed, red being lowly or non-detected. If we then parse out, I don't have time to go into all of the differences, but if you look below there in E, you can see that if we specifically look across the interpersonal violence groups, there's this cluster of blue here, these highly, highly abundant proteins only detected in this one group, adolescent sexual trauma group. And if I show you what those are, it's quite surprising. In the center here of this dot plot, you can see there's a large number, something like 27 of these proteins that were largely abundant and only detected in women who'd experienced sexual trauma during adolescence. And they were all related to keratin and keratin-associated proteins. This took me back to my postdoc days when I was studying the stress axis, where we were reminded that the stress axis that exists in the hypothalamus is very similarly replicated in the skin. And interestingly, these keratin, all these genes, the majority of the ones we detected in circulation, the genes themselves were all part of a 17q21 gene cluster, meaning on chromosome 17, on this arm of the chromosome in this cluster that has previously been identified, that the majority of these genes were part of this cluster. Also of interest, as you can see here, CRF receptor 1, so a stress-related receptor, and MAPT were also identified in this cluster. And these genes have also been identified in the GWAS, being important for PTSD risk. So in summary, isolating in an unbiased approach EV proteins, we were able to identify a unique location within the genome. And gene clusters are extremely exciting because gene clusters tell you that there may be a time across the lifespan at which that cluster, being epigenetically regulated, was vulnerable to some change in the environment. So we think further about what we learned about these genes is that the majority of them were super interesting because they pointed to the differentiation of keratinocytes in your skin into Merkel cells. And you may remember from a few years ago, the Nobel Prize was actually given for the piezo-2 Merkel cell discovery by Artem Patapoutian and others. These piezo-2 Merkel cells, piezo-2 positive Merkel cells, are mechanosensory skin cells. So they respond to light touch, and they contain piezo-2 channels as the transducer, which when activated release norepinephrine and other neurotransmitters. So they are the only neuronal-like skin cell. And they're interesting based on the timing I just showed you and the cluster that I just showed you, that their final maturation of this differentiation occurs in adolescence. And so I just pointed out some really exciting papers here if you're interested in this process. But it seems fitting to us that a particular type of trauma, sexual trauma, because of where these Merkel cells are and when they are differentiating. Merkel cells are in your fingertips, they are in your lips, and they are in your genitals and particular places on the back of your skin. That this particular type of trauma may have programmed within this cluster the differentiation and responsivity of these Merkel cells, leading to their detection, their protein detection in EVs from these women. So I just wanted to highlight from this unbiased approach, going back to the G by E by D, that we're able to isolate a unique gene cluster within chromosome 17, that we're able to identify the particular type of trauma experienced, the E, as well as the timing of the developmental window of adolescence. So we are now continuing on a grant funded by the National Institute of Mental Health, with Tanya doing a particular assessment in another group or cohort of individuals that includes men and women, where their mothers were recruited in pregnancy 34 years ago. And these children have been followed for 34 years. So we know all of the exposures and experiences that they've had. So we can again isolate particular types and timing of trauma. And they're now being recruited back in. They're all 34, 35 years of age. I've met with many of them in a community event that we held a few months ago, which was incredibly exciting to include that community into the type of research that we're doing and assess the unique role that these Merkel cells may play in trauma and perhaps in prevention and in treatment. You can think of lots of ways in which the Merkel cells and perhaps types of therapies could be involved in PTSD. So what has this led to mechanistically? So in this grant, we are also looking using the preclinical mouse model, where we can specifically identify these Merkel cells as being part of the stress axis, identifying how they're involved. One of the ways we're doing this is laid out here using DREADS. For those who don't know what DREADS are, the DREADS were developed by Brian Roth at UNC, and they are designer receptors. They're G protein coupled receptors that are exclusively activated by designer drugs. So they're G protein. We use the ones that are specifically coupled to GQ. So when activated in unique cells, so you can see there that we can use Merkel cell specific Cre transgenic and cross them with a conditional DREAD GQ. So now we're putting unique G protein coupled receptors only in these Merkel cells and Merkel cell enriched populations. And we can do all sorts of things by doing ways of treating these animals with, I'll show you in a second, the drug is called CNO, and we put it into cookie treats. So we can activate locally these cells and ask, what changes does it do to their stress reactivity, to long-term trauma programming, et cetera? And I'm going to show you some data from this in a minute. So the first question we had to ask when putting a novel receptor into Merkel cells and activating it, because we can't have a conversation with the mice, is to ask, what does it feel like? Is it a positive or negative outcome to have your Merkel cells activated? And so I'm just going to play 2 quick videos here. These are either dread positive or dread negative animals. Everybody just got this cookie treat containing 2 mg per kg of this CNO drug. And I'm going to ask you to try and guess which one here are the dread positive animals that are having their Merkel cells activated and their behavior. So the first thing, if you don't work with mice, you need to know is that when mice are moved, so you can see these cages were put into the hood. They're very curious. They'll explore. When mice are stressed, their first response is to go back into their nest and groom. So I'm going to activate both of these videos. So you can see on the left is one genotype and on the right is another. And so you can see very quickly the animals on the left are exploring their environment. All of the animals on the right, you can see, are back in their nest and massively grooming. So it won't surprise you to find out that the animals on the right are the dread positive animals, right? So all animals got the drug. Only the dread positive animals, though, are responding. So it's likely that they notice that these skin cells have been activated. We've done follow-up studies. It doesn't seem to be a positive association or a negative, really. It's just that they notice it. And the first thing that mice do, because they're very stress reactive, is to groom. Okay. So because we are running short on time here, I think I'm going to skip to the end of this second part of the story, because I know you have a program that you need to attend to, and I want to leave time for questions. The second part of the story was really going to go back into a preconception environment in question. And this is focusing on the preconception germ cells, both mom and dad. And I was going to tell you a story about dad today and trying to really understand with a very specific type of cell, the sperm, and how in mice in a preclinical question and humans in a clinical question, experiencing stress in your environment can shape the function of these cells and the cargo that these cells carry to conception that ultimately shape the rate of development of the brain. Unfortunately, I don't have time to get into this, but there's lots of history around the world of how traumatic events have altered outcomes in brain development. We've published a ton of work over the last 2 decades assessing when and how in preclinical studies and in cell culture studies, these effects. I'm going to summarize this in one slide basically here to say that it's not during the course of spermatogenesis that the environment shapes the sperm, but rather when sperm leave the testes and spend a few weeks in the epididymis. You can see the tubules here in cross section. These cells are called epididymal epithelial cells. And these cells secrete all kinds of important factors, including extracellular vesicles, that are essential for the final maturation of sperm. And that's important because you can now imagine a way in which adversity in the environment, a pandemic, famine, war, can have lasting effects on these somatic cells that are so critical for the signals and the final maturation these important germ cells then carry to the egg at fertilization. And again, here's all of the work. Many individuals in this field have studied many aspects. We do stress in our preclinical model, but many other individuals around the world have done studies on related to famine and other types of stress in the environment. I don't have time, unfortunately, to get into all of the mechanistic studies we've done, but this was the thesis work of Jennifer Chan, a graduate student, while I was at Penn. This is all published if you're interested, but we've done all sorts of methodological approaches to try and understand what are the signals being relayed to the sperm, how long do they last, and then how do those sperm changes affect fertilization, implantation, and the brain's development, all of which is very important for understanding the brain. And so I also want to highlight here that understanding mechanistically how these effects happen, there's many ways to do this, but understanding causality as a neuroscientist, I think, is important. So we've done this by doing sperm comparisons of secreted EVs from those epididymal epithelial cells, implanting them after performing intracytoplasmic sperm injection. And we can study by looking in the uterus how the brain develops and letting those animals grow up and ask about how they interact in their environment. This is all work from Jennifer Chan's thesis that is published, but it just highlights there are huge differences in the process of neurodevelopment just from changing the interaction of sperm with those EVs from stress. And it ultimately not only changed the rate of the brain's development, but how those offspring then interacted in their environment later when they were adults. So I'm going to leave you as a neuroscience community with a thought about this germ cell that is so important coming from Dad's experience, that if you think about what a sperm is, and you wanted to build a better sperm for fit in an environment that has experienced a lot of stress and trauma, you would need to appreciate what is sperm. So sperm is really just Dad's DNA that needs to be brought to conception and the motors that drive that DNA to get there. So one of the factors the environment can influence to make a more efficient sperm would be affecting those mitochondria that produce the ATP for the motors to get the dad's DNA to the egg. And sure enough, we've done this preclinically now and clinically to assess how stress through these EVs as laid out here, and this work is now published just recently in Nature Communications, also from Nicole Moon, who was an MD PhD in the lab, and now she's back in the clinic. But just to show you that we change the motility rates here by exposing the sperm, these are mice, to the EVs from stress. We increase all aspects of the rates and velocity of the sperm to reach the egg. And this is largely done through shaping the outcomes of the mitochondria and their respiration rates, which I don't have time to get into. So I just want to leave you with the fact that the preclinical model and our 15 years of work in this area has now led to several studies in humans in partnership with Neil Epperson, who is the chair of psychiatry here. But Neil and I were both working together on these studies while at Penn as well. And so this work is now published. I'm not going to spend time on it, but we found lots of changes in relationship to Penn undergrads, actually. Daniel, if you're interested, in assessing outcomes over the course of time and the composition of males' sperm in relation to their perceived. And so we have now taken those studies here also in Colorado, following up on another grant from NIMH that Nicole was working on in men also, and understanding how life adversity and stress and seeing the same change. I'm showing you on the right here. In the month of the perceived stress, there's no change in the motile rates of the sperm, but at time following, which goes again to the maturation rates that I showed you earlier, that you start to see this one-to-one relationship for the level of perceived stress increasing the motility rates. And what we're ultimately seeing in these studies is that this timing supports the fact that the rate at which and the efficiency of the sperm to reach the egg, the cargo the sperm are carrying, leads to better implantation rates and a faster rate of embryo and fetal development, all of which is so important for understanding for the brain's development itself. So we've now taken these studies, going back into the mouse and doing all kinds of assessments with live action microscopy and understanding exactly how and what parts of the cargo are affecting that rate of development to the implantation. So just summarizing all of the things I just told you here and understanding the roles of extracellular vesicles and how these nanoparticles are so important at the level of circulation as biomarkers, understanding how over life's adversity and trauma and stress, this really becomes a wearing down of the cell's ability through all tissues to maintain homeostasis. You can imagine then how that affects your vulnerability for mental health disorders. And even at the level of specifically asking what these EVs communicate, at the data that I didn't have as much time to talk about today for germ cells, but how important they are as cellular communicators and cargo is something that we're currently pursuing for the developing brain. So ending there with, again, thanking Anna, Daniel, thank you for organizing this session. I hope you have a fantastic meeting. Again, giving a shout out to Falan and all of the great work that they do to coordinate neuroscience in Latin America, to all of the IBRO regional committee members who are there. I hope that you all have a fantastic meeting and that you all will be big advocates for the work that IBRO does around the world, for trainees, lifting them up and providing access to training experiences, travel, etc. And so thank you all. And if we have any time, I know this has been a little difficult. I'm going to stop sharing here. A little difficult, but I want to make sure that there's a time for any questions that anybody has. Thank you all so much.
It was wonderful. I apologize for the technical issues, but we were able to understand you for sure very well. So we might have time for quick questions if you have. There. Are you listening?
Hi. Hi. I was wondering about the differentiation of the Merkel cells, if it maybe could be explained partially the enhanced sensitivity that the women who experience post-traumatic stress after sexual assault have, like they are very against people touching them, and if it may be related or not, because perhaps the time frames are do not coincide.
So that is a fantastic question. I love your thinking. We've spent a lot of time exactly in this space as to why we're detecting these EVs in circulation and only in this group. And that's a fantastic way of thinking about it, because in order to get the blood sample, right, they're recruited in to do the fear conditioning and startle, and they're doing blood sampling. Well, of course, we all know to get a blood sample involves a stranger probably having a lot of contact with you. And so is that because EVs are so dynamic? Is that part of the process and why we're detecting them? If you recall, microvesicles themselves, the specific EV that blebs from the membrane of a cell, so for instance, a Merkel cell, they are very fast in how they're responding. And so it's a really interesting idea to think about that the 17q21 cluster in the differentiation of these cells that may have occurred during this window of time change their sensitivity such that now when those blood samples being drawn, that stressor is containing an abundance of EVs from this cell type that is not found then in the other groups. So it's sort of the why and the when. But yeah, I really like that thinking. And it's something that we're exploring more in our current cohort because we're getting a stress reactive stressor that involves sensory modalities to get a before the sensory stressor and after. And will include sex as a biological variable, so also exciting.
We are running out of time, and of course of your time, your generous time. So Tracy, I will thank you again, and please join me in a big applause to Tracy for her wonderful talk.